Okay. But I would like to see me on the screen. Full. Live feed. Where is it? On the floor? Okay. I can talk now?
Yes.
Live feed. Now I see it. Okay. Yeah. Yeah. Good morning, good afternoon, and good evening, everybody. Welcome to the session Related Diseases. I'm Michael Beck, geneticist and pediatrician, and I'm involved in lysosomal storage disorder for almost 40 years. And I was involved in I was the head of the Center for Lysosomal Storage Disorders at the Children's Hospital in Mainz, Germany, but I am now retired. And I will be the moderator for this session. Cause facial features, organomegaly, skeletal abnormalities, and neurodegeneration raise the suspicion of a mucopolysaccharidosis. But there are, however, other lysosomal storage disorders that present with signs and symptoms more or less similar to those of mucopolysaccharidosis disorders. And these conditions are the subjects of our session. The keynote speaker, Dr. Mengel, will give an overview about similar diseases. Professor Braulke von Hamburg will talk about mucolipidosis, and Professor Dazzo from United States will talk about pathogenesis and therapy of sialidosis and galactosialidosis. The keynote speaker, Dr. Mengel, joined my institution in Mainz in 27 years ago. And there he worked as a consultant for metabolic disorders in general and was principal investigator for many, many lysosomal storage disorders. 2 years ago, he left the Villa Metabolica and founded a private institute in a small city close to Frankfurt. And this institution is called SINGS, Sphingolipidosis Clinical Science. And here at this institution, as a head of this, he is performing investigator-initiated studies and other multi-center studies in the field of lysosomal storage disorders. In all these years, he has got huge experience in all lysosomal storage disorders, not in all the common, but also in very rare conditions. And now I will give the floor to Dr. Mengel. Please, Eugen.
Michael, thank you very much. I felt really honored to be invited on to give a lecture about related diseases. And what's the understanding of related diseases? We understand that there are several lysosomal conditions which are similar to mucopolysaccharidosis. Similar in terms of clinics, in terms of pathophysiology, and last, and that's a very pronounced topic of my talk regarding treatment approaches. But these lysosomal storage disorders are not per se mucopolysaccharidosis. Similar in clinical manifestation means these disorders have, like MPS, cause phaseus, they have macrocephaly and dolichocephalus, and they may also have short stature. Common pathophysiology means also substrate accumulation, lysosomal dysfunction, and downstream pathology. And most important, related diseases may also have similar treatment approaches in terms of intravenous enzyme replacement therapy, intracerebroventricular enzyme replacement therapy, substrate reduction therapy, chaperone therapy, and last but maybe in future most important, gene therapy. The related disorders can be divided in different groups according to the substrates in these disorders. It can be related, one group is the sphingolipidosis, for example, GM1 gangliosidosis, also GM2, acid sphingomylase deficiency, sialidosis, and galactosialidosis. The group of mucolipidosis, especially mucolipidosis types 2 and type 3. I listed here not mucolipidosis type 4 because it remembers me not in the first line on mucopolysaccharidosis. It remembers me this disorder on steroid lipofuscinosis. There is a group of glycoproteinosis, alpha-mannosidosis, beta-mannosidosis, fucosidosis, and aspartylglucosamminuria. And there are some other lysosomal storage disorders which are difficult to group according to their substrate. One example is free sialine acid storage disorder. Clinically similar means in the first hand, the typical sign of MPS is cos-facial features. It's part of GM1, gangliosidosis, sialidosis, galactosialidosis, also mucolipidosis type 2, but it's maybe not very much known that cos-facial features can also be part of the infantile form of free sialine acid storage disorder and acid sphingomylase deficiency. I will show on that. Bone dysplasia, it's here in some of the related diseases, a common feature, and also in all related diseases, early onset of neurodegeneration is a clinical sign. And lastly, clinically similar is macrocephalus and dolchocephalus in some of the sphingolipidosis, in mucolipidosis, but also in this very rare glycoproteinosis and also in infantile free sialine acid storage disorder. Cause features are maybe indistinguishable from MPS disorders, especially in GM1 gangliosidosis, also in mucolipidosis type 2. But maybe not the common, the whole picture of cause-facious features of MPS is seen in free sialine acid storage disorders. Here in this girl published from Hulsing in this year, very new picture about this. With the intermediate phenotype, you see the settled nose, the prominent forehead. So some features, not the whole pictures. And that's also true in acid sphingomylase deficiency. Here published from the Czech group in 2005. And interestingly, these cause-facious features in ISMD are mainly associated with one common mutation, Q294K, and that's a common mutation in East Europe and Middle Europe. Nobody knows why, especially in these patients, you found the common cause-facious. For the diagnostics, clinicians think often on lysosomal storage disorder, MPS, and related diseases. If cause-facial features are associated with cognitive decline. I give here a list of diseases where the combination of cognitive decline and coarse facial features reflect the manifestation of the disease. And you can see the decline, the cognitive decline in these patients, in these disorders, is very heterogeneous. It's maybe mild to moderate for aspartyl-glucosamminuria, but on sialidosis, it can be very severe, especially when it combined with epileptic encephalopathies. A further common finding in MPS disorders is corneal clouding. It's a clinical manifestation of all the diseases, with some exception in Hunter's disease. The grade can be mild opacities, but in some patients, especially in the more severe phenotypes, over time you find a complete opactic cornea. But there are also, it's common in the related diseases also. In some of them, it can be as severe as in MPS disorders like GM1 and sialidosis, but in other disorders, it's maybe mild, and you have to look with a spalt lamp to make the diagnosis of corneal clouding. Even a common finding, the most severe biologic catastrophe of manifestation of a lysosomal Stars disorder is non-immune hydrops fetalis. It means that usually the patient diseased in utero. It's common in MPS 7, but it's also part of the clinical spectrum of galactosialidosis, of Gaucher disease, of mucolipidosis type 2, sialidosis, free celiac storage disorders, and GM1 gangliosidosis. All these disorders, despite different substrate, despite different protein deficiencies, have clinical similarity. It gives us the idea that there is a common pathophysiology behind this disorder. And there are two papers around which reflect this common pathophysiology, and additionally, it's so that sphingolipid storage is a pathological finding in MPS and in related diseases. It's shown by Konrad Santow, and here is the picture from him, the degradation pathway of the sphingolipids, and the first one here is GM1 gangliosidosis, and the last step is the ceramidase deficiency here. And what Konrad Santow and Ms. Brighton are announcing is that in all this disorder, not only in the gangliosidosis, GM2 and GM3 storage can be found. It can be found in a number of mucopolysaccharidosis. It's shown more than 10 years ago from Steve Walkley and Marie Vanier. They summarized this in an excellent review. But it's also shown in non-MPS diseases. It's shown in Niemann-Pick diseases that from Ceres, that's GM2 and TM3 is part of the pathophysiology in mucolipidosis type 4, in ceroid lipofuscinosis, and also very early, it's 20 years ago, it was shown for alpha-mannosidosis. And so GM2 and GM3 is maybe a therapeutic target for not only for sphingolipidosis, also for related disorders. Ms. Pryden and Konrad Santhoff had shown this in detail, where GM2 and GM3 is found. They make an excellent review and give reference if you, to this common pathway. And in the end, they discuss pathophysiological model, so that the lysosomal enzyme is in the beginning of the disease. It's causing primary storage, but then also secondary accumulation, especially of GM2. And downstream, it's not the end of the pathophysiology. So in these disorders, also autophagy, inflammatory response, cellular damage, accumulation, it's a model which fits with all of the related diseases. And the second paper I like to mention is from Gianni Parentini and Andrea Palladio. In the beginning of the paper, they demonstrated the understanding of lysosomal storage disorder, starting with the first description of Tay-Sachs disease more than 150 years ago. And in the end, the detection of the genes and the first introduction of treatments. And in the end, they give also perspective what's here. And similar to Konrad Santhoff and Annette Pryden, they also demonstrate a pathophysiological model with primary storage, with secondary storage, impaired autophagy, mitochondrial dysfunction, and altering of signal pathways and inflammation. And they thought this pathophysiological model of lysosomal storage disorder has also implications for therapeutic approaches. So if gene therapy works in one disease, it may work also in the other disease. And there was a big discussion that, and we saw it in the last days also here, that adeno-associated viral vectors, as well as lentiviral vectors ex vivo, are the most common approaches to target neurodegeneration. They also discussed intrathecular and intraventricular delivery of enzymes. It has proposed for mucopolysaccharidosis as well as for metachromatic leukodystrophy. Here for metachromatic leukodystrophy, the publication was last year, and now we have also this therapeutic approach for the NCL type 2, which this publication on ceroliponase alpha from Angela Schulz 3 years ago. And also their therapeutic approaches common in related and MPS disorders are the use of chimeric lysosomal enzymes, diffusion proteins, may cross the blood-brain barrier to antibody components that receptor mediated, and that will be used in future, also not only in preclinic. It will come to clinics, that's for sure. Therapeutic approaches who are now also in clinics is substrate reduction therapy. It's not on, it's may focus on sphingolipidosis, and the principal way is to stop the first step of ganglioside synthesis by the inhibition of glucosylceramide synthase. And it's investigated in the past for GM1 and GM2 gangliosidosis, and but in future, and I will look to that small molecules, new small molecules are maybe more effective than miglustat. Pharmacological chaperone therapy, that means that a small molecule causes stability of the mutant enzyme and prevents the mutant enzyme for intracellular degradation. And in the end, a chaperone may give some more protein and enzyme function. The model of this is also published some years ago from Giancarlo Parenti. But this downstream pathology gives also some ideas of new adjuvant therapies to target downstream pathology. It's a long discussion already, and we have heard in the last days about it, to alter pathologic autophagy to regulate GFAP, but also from Lila Simonaro and at Schuchman, some work is done on pentopolysulfate, and it's on this medication targets an anti-inflammatory effects on lysosomal storage disorders. In the beginning, it was thought only for MPS 1 and 2, but also it's investigated actually for MPS 3A and MPS 6. And there's another very interesting small molecule around, it's arimoclomol. It's now in clinical trials with Niemann-Pick disease type C. And this small molecule effects on Hitchcock protein 70. It stimulates this protein genetically, and this has some effects on downstream pathology, on impaired autophagy, in lysosomal stability, and also on protein degradation. All these therapeutical approaches were in a recent publication discussed in Fabry disease. The colleagues from the AMC in Amsterdam around Kolle-Hollack, they have very nicely illustrated therapeutic approaches which are used and in clinical research on lysosomal storage disorders. It's bone marrow transplantation, it's enzyme replacement therapy with per infusion or per bone marrow transplantation. Discussions on gene therapies, we have heard in the last days on that. and also substrate reduction and chaperone therapy. It's very nicely explained and discussed here. And on the related diseases, we have the alpha monocytosis, and 2 years ago it was approved for treatment, intravenous enzyme replacement therapy. But what we learned from alpha monocytosis is that that has a big limitation of intravenous enzyme replacement therapy. It's the blood-brain barrier. Such protein have difficulties to reach the brain and may not change the neurodegenerative cause of the disease. Chaperone therapies are since several years discussed for free-sialic acid storage disorder. It's the thinking that a chaperone may stabilize this protein and gives the transporter to the lysosome some function in vivo. In aspartyl-glucosamminuria, also some years ago, it was discussed from the Bunning group here that chaperone therapy is maybe a way to treat, but they recognize that the mutations in aspartyl-glucosamminuria may not cause protein misfolding and early protein degradation. They cause nonsense-mediated decay. And there's a recent publication here from 2018 that amaloxone could be a candidate, at least in preclinical studies. It has shown some effect on increasing enzyme activity by blocking nonsense-mediated decay. In the last part of my talk, I discussed on beta-galactosidase deficiencies. It's there are 2 disorders known. One is classical MPS, is MPS 4A, with keratin sulfate storage and skeletal dysplasia and corneal clouding. And there's GM1 gangliosidosis, sphingolipidosis, which lead to a very severe neurodegenerative disorder, but has also some aspects on skeletal dysplasia. It's GM1 gangliosidosis divided in 3 types. Of course, it's a spectrum, and the types are maybe artificial, but in clinical routine, we divide it in type 1, the infantile form, type 2, late infantile and juvenile, and type 3, the adult form of the disease. And here again, the common pathophysiology behind. And for me, in personal, very important paper 2 years ago from the Biomarine group in cooperation with Alessandro De Idiazzo. They investigate in the mouse model of GM1 gangliosidosis, intracerebral ventricular enzyme replacement therapy, and they show very nicely some positive preclinical results. But I have the impression they look now what are doing the other approaches in GM1 gangliosidosis. And one of the approaches is substrate inhibition therapy. And nowadays in very near to clinical trials and even in clinical trials are 3 substances: Venglustat, Azazucar from Azavaro, and Simplacostat from Idorsia. Venglustat is from Sanofi. And the first small molecule which was investigated in GM1 gangliosidosis was lucerastat. It was about 15 years, 16 years ago, and they even show some promising results, but maybe the brain delivery of lucerastat is not optimal. But they could see, they could histologically see that treated mice are in between diseased and controls. More recently, vanclostat is coming in clinical trials for sphingolipidosis, for neuronopathic sphingolipidosis, and the basis, the preclinical basis, is this paper here from Marshall and the colleagues from Sanofi Janssen. And what you can see in the mouse model of GM2 gangliosidosis, a survival of 30 days more. And they could also show some improvement in weight, some improvement in neurological manifestations in the mice, and also some manifestation in histology, and some changes in a biomarker that's where maybe becomes important in the next years in lysosomal storage disorders. But what we all seen here is that the treated are something between the more severe form and the unaffected. So it's always in the middle. The same is maybe true for this azosugar, this biphenyl-substituted immunosugar, published first from Hans Arz and colleagues in Leiden and earlier in Amsterdam. It was, and they had shown that is also not substrate reduction by glucoceramide synthase. There are also this substrate, this small molecule also target neutral glucosylceramidase, which is involved in by glucosylceramide degradation in the cytosol in the pathological pathway. So very important. And on the third immunosugar, sympaglostat, the animal model is not published so far, but some informations are seen on the Idorsia website if you have a look. The other big approach in clinical trials is it's gene therapy for GM1 gangliosidosis. It's from 3 companies. The first one is from CEO, and that's I will say the classical AA49 intravenous infusion for therapy, and here the patients who can take part in this but are not in more severe forms. It's type 2, and that's maybe the question mark behind is intravenous infusion by an IFU. How much is the correction by enzyme replacement therapy? And you may need in these patients not as many corrections as in infantile forms. Yeah, and you see also this, it's lady infantile, older than 2 years, so that's not the classical infantile phenotype. That's different in the studies by Passage Bio. They deliver also an AAV vector to the cisterna magna, so near to the brainstem. And it's a vector with a serotype, very uncommon, very special, U68. And the third company, it's known in the fields and since several years, it's Lysogen. They used also an EAV vector here, RH10, and it's also, and they looked how it works in the first step on 4 subjects with infantile GM1 gangliosidosis, and they named it adaptive design. So they will look how it works here and then design further on. Very interesting. So coming to the end, I like to conclude. Related diseases can be clinical, similar, and important differential diagnosis. But the most important is that they have a concordant pathophysiological features, the lysosomal steroids to appear, and that the spectrum of therapeutic approaches is consequently also concurrent. And on the both big groups of related disorders, mucolipidosis type 2 and type 3, Thomas Brauke will report shortly after me, and Alessandro Te also follows on sialidosis and galactosialidosis. And I will enjoy these talks. Thank you very much.
Thank you very much, Dr. Mengel. And it was a very good overview. And one is surprised that related diseases are so common. And that you have emphasized that these diseases are not, do not resemble mucopolysaccharidosis from the prospect of clinical manifestation, but that they have also pathophysiology and treatment in common. I hope there will be some questions. Eugen, before I see the questions, one question you have mentioned, but you did not give go into detail, GM2 gangliosidosis, and also in this field one is considering gene therapy. But for Tay-Sachs, for example, you would need 2 genes to incorporate, and I think 2 genes in one AAV vector would be too large cargo for this virus. And do you have any idea how Tay-Sachs patients could be treated with gene therapy regarding the 2 genes that may be involved in this disorder?
Yeah, in comparison to GM1 gangliosidosis to GM2 gangliosidosis, there are also with comparable vectors and techniques than in GM1 gangliosidosis. But the issue is that you have for the GM2 alpha and a subunit in this enzyme. So in the end, you have 2 genes which are involved in the diseases, and how to balance the cooperation of the subunits here. So that's a very critical point. And there are some publications around, preclinical publications with gene hybrids which are edited by gene editing, and it's called HEXM. So it's an artificial enzyme which acts with the activator and gives, at least in neurons, function back. So a very interesting gene editing approach for GM2 gangliosidosis. Yeah, and we have to look in future what will happen here on the preclinical research basis.
Okay, that means that there's a hope for GM2 gangliosidosis too for gene therapy.
Yeah, there's even some trials started, to my knowledge, from 2 companies, and there is preclinical research on new gene therapy concepts.
Okay. There is several questions I must see. Question. One question is in Spanish. I can understand a little bit Spanish, but I need a translation of ensayos. What are ensayos? Dr. Ramon Canabal Pombo has asked this question in Spanish. He or she talks about a patient with type 2 GM1 gangliosidosis and asks for participar en los ensayos.
It means studies.
Yeah. Okay.
Clinical trials.
The question is GM1, type 2 GM1 gangliosidosis, whether he or this patient could participate in a clinical trial.
Yeah, in general, clinical trials are listed on the homepage of clinicaltrials.gov, the study overview of the FDA, and on this clinicaltrials.gov, there are inclusion and exclusion criteria listed, and in the most trials there's also a contact person from the company who offers the trial, or from the principal investigators who are performing the trials, and that should be the way to contact and ask for study participation.
Okay, thank you very much, Dr. Mengel, and I hope this answer helps the participant. Now we'll go to the last presentation that will be given by Dr. Alessandra Dazzo. Dr. Dazzo graduated in biology and was a doctor in genetics at the University of Milano. And thereafter, she worked at the Erasmus University in Rotterdam. And after training, she had the opportunity to work some time with Dr. Elisabeth Neufeld at the end. And thereafter, she came back to Erasmus University in Rotterdam. And now she is a professor and chair for Department of Genetics, St. Jude Children's Hospital. And the title of her presentation is Sialidosis, Mucolipidosis type 1, and Galactosialidosis, Pathogenesis and Therapies. Please, Alessandra.
Thank you very much, Michael, for this kind introduction. Good morning, good afternoon, and good evening for everybody who is attending. I'd like to start by thanking the organizer for giving me the opportunity to participate to this interesting and very instructive meeting. And I also want to extend my thanks to the people who are organizing this virtual meeting, especially David and Julia, who had been running it in a very smooth and efficient way. So thank you. Today, I will talk about 2 diseases as already anticipated by Eugene and Michael. Sialidosis and galactosialidosis. I will touch upon briefly our studies on the pathogenesis of these diseases and testing of a number of therapeutic interventions on mouse model of these disorders. But let me start with this concept. I think is now a consensus, I believe, among investigators in the field of lysosomal storage diseases, that LSD can be considered diseases of early aging. This concept stems primarily from a number of observations made in faithful models of LSDs that share pathologic features of adult age-related diseases. These animal models have been extremely useful for investigating mechanisms of pathogenesis that are apparently common between pediatric LSDs and adult diseases, especially neurodegenerative diseases. As you heard from the many interesting and eloquent talks we heard so far, the pathogenesis in LSDs is often linked to the accumulation and often abnormal redistribution of undigested metabolites or substrates or storage downstream of the primary defect in cells of multiple tissues and organs. And studies of the pathophysiology of these diseases in animal models have brought to the identification of many pathways that are directly related to lysosomal dysfunction, so are regulated by a proper lysosomal function. And this common pathway found dysregulated in both LSDs and many adult diseases include, as you heard, autophagy, endophagocytosis, calcium signaling, oxidative stress, lysosomal exocytosis, inflammatory and immune responses, and different forms of apoptosis. My lab has been working for many, many years on 3 diseases that are linked to primary or combined deficiency of 3 lysosomal enzymes that assemble in a high molecular weight lysosomal multi-enzyme complex. The enzymes are the lysosomal sialidase NEU1, the lysosomal acidic beta-galactosidase, and one of the lysosomal catepsins called the catepsin A or protective protein catepsin A. These 3 enzymes highly depend on each other for their correct processing, compartmentalization, acquisition of full enzymatic activity, and stability in the endolysosomal system. So if we want to properly address the pathophysiology and therapeutic intervention in the 3 diseases linked to these enzymes that I will discuss in just a few minutes, we really need to take into account the fact that the 3 enzymes form this high molecular weight complex. Today, I will focus primarily on 2 of the 3. Neuraminidase 1 is a canonical sialic acid cleaving enzyme, is one of 4 mammalian sialidases. The preferred target substrates of these enzymes are glycoproteins, but the enzyme can cleave glycolipids with the aid of detergents, of activators. The substrates, several substrates, target substrates have been identified, but I think we are all at the tip of the iceberg in terms of the identification of the many substrates that lysosomal enzymes actually target. Quite few of them are lysosomal membrane proteins, including LAMP1 and LAMP2. We identified the amyloid precursor protein, several cell surface receptors, including the insulin receptor and many others. But also, as I mentioned earlier, the direct target of Neu1 are also the partner of the complex, PPCA and beta-galactosidase. A primary deficiency of Neu1 leads to sialidosis, a glycoprotein storage disease. And the storage material is primarily sialylated, or are primarily sialylated glycoprotein and oligosaccharides. Going to the catepsin A is a carboxypeptidase that is acidic pH, but also maintain a deamidase and esterase activity and more neutral pH. The substrates identified that for this particular catepsin are several bioactive peptides, including many regulating the blood pressure, like endothelin-1, and LAMP2, which is, as you all know, the receptor of chaperone-mediated autophagy. A primary defect of PPCA leads to a secondary severe deficiency of Neu1 and partial deficiency of beta-galactosidase, and the disease is called galactosialidosis. Some of the first patients were actually diagnosed by Michael Beck. It's a disease is also part of the glycoprotein storage disease. And because of the secondary deficiency of Neu1, galactosialidosis storage is primarily sialylated oligosaccharides and glycopeptides, basically identical to the one seen in sialidosis. And this is one of the several similarities between these two diseases, but I will also stress that there are several differences between the two conditions. So sialidosis and galactosialidosis, I don't need to go in details since Eugene had been very explicit in discussing some of the major clinical features. They are bona fide lysosomal storage diseases. They present with a continuum of disease severity. Often enough, many of the characteristics of the pathophysiology and pathological features are shared by galactosialidosis and sialidosis, especially those patients that are diagnosed with the early infantile form of the disease. But as you see, both in galactosialidosis and specifically sialidosis, there are also attenuated forms like the late infantile galactosialidosis that is practically non-neuropathic, and sialidosis type 1 that is normomorphic and is also referred to as cherry red spot myoclonus syndrome. Again, the hallmark of the and diagnostic hallmark of the disease is oligosacchariduria that is shared, as I said, by the two conditions. The mouse models we have generated a long time ago are really quite faithful models of the severe forms of these diseases. They present with a very severe systemic condition affecting visceral organs, but also bone, cartilage, muscle, and of course, invariably the nervous system, the central nervous system and the peripheral nervous system. And looking specifically at the characteristic of these mice and in search for pathogenic mechanism that would explain specific phenotypic alterations in sialidosis and galactosialidosis, we stumbled upon potentially important pathways that is directly downstream of neuraminidase. And neuraminidase is actually a negative regulator of the general pathway called the lysosomal exocytosis. Lysosomal exocytosis is a calcium-regulated process that is actually used by many cell types, if not all cell types, to actually repair the plasma membrane. It entails the recruitment of a selected pool of lysosomes along the cytoskeletal network to the plasma membrane, where they dock prior to fusing with the plasma membrane. and releasing their content extracellularly. And I talk about content in terms of soluble lysosomal hydrolysis, as well as a particular fraction of the lysosomal content, including exosomes, for instance. The docking step of the pathway is very dependent on LAMP1. And interestingly enough, LAMP1 turned out to be one of the major targets of Neu1. In absence of Neu1, LAMP1 remains sialylated. And because of an increased number of lysosomes decorated with a sialylated LAMP1 at their membrane, a number of lysosomes dock and at the plasma membrane, ready to engage in lysosomal exocytosis. The end result is a massive release of active hydrolytic enzyme extracellularly, with all the consequences that entails for the extracellular milieu, and a redistribution of LAMP1 at the plasma membrane, which is now became a proxy for identifying excessive lysosomal exocytosis in different cell types. So following this paradigm, LAMP1 sialylated and of course caused by Neu1 deficiency, LAMP1 sialylated, excessive exocytosis, release of soluble hydrolysis and exosome into the extracellular space, we actually relate this aberrant or the deregulated pathway in the pathogenesis not only of sialidosis, but also of disease of other adult conditions, most of them of unknown etiology, like sporadic Alzheimer's disease, idiopathic pulmonary fibrosis, and cancer. This discovery offers, in fact, a new therapeutic option for the treatment of LSDs and also the possibility to exploit the lysosomal enzymes therapeutically for more common adult diseases. So let me give you a few accounts of what we have seen specifically in the brain of the neuraminidase deficient mice. The mice accumulate abnormally very early in life the APP precursor protein in the endolysosomal compartment, as you see in this punctated staining. But as the animal age, the APP positive proteinaceous materials started to accumulate in the form of plaques, especially in the hippocampal region, and especially the CA3 regions of the hippocampus of these mice. As I mentioned, we found that APP is actually a substrate of Neu1, remains sialylated in absence of the enzyme, and as a consequence, abnormal accumulation of APP in lysosomes result in abnormal toxic cleavage of this precursor protein into toxic peptide, including amyloid A beta, that is then released extracellularly by lysosomal exocytosis. And this is seen both in primary neurosphere cultures, in the medium of primary neurosphere cultures, as well as in the CSF of these mice. Last but not least, interestingly enough, at the ultrastructural level, the histopathology of Neu1 - hippocampus resembles very closely what is known to occur not only in canonical AD models like the one shown here, but also in AD patients. And that includes the presence of dystrophic neurite, full of autophagosomes and lysosomes, as well as neurofilaments. And these are features that are completely reproduced in a canonical AD model. So we can consider Neu1 as presenting with an 1 mice as presenting with an AD-like phenotype. Most importantly, keep in mind that, of course, I don't have time to go in details to this, but this particular accumulation and phenotypic alterations give rise to massive neuroinflammatory response that is completely superimposable to what is known to occur in AD patients. Importantly, we have started the assessment of a number of sporadic AD sera, where we see that with the increment age of the patients and BRCA severity of the disease, we see an increment of beta-X activity in the sera, again, as a proxy for at least a preliminary indication that they might have an increased lysosomal exocytosis. And the same holds true for the CSF of AD patients. And I want to show you briefly the extensive APP accumulation in the brain, clear brain, of U1 mice. As you see, this shows the entire hippocampal region. The accumulation starts in the CA1 region and spreads throughout the CA3 in this large puncta. If we compare this with what is happening in the PPCA minus mice, we notice that there is a clear difference in the accumulation and expression of APP in these mice. You see a much finer puncta in the CA1 region, and this accumulation in older mice never reach the CA3 region of the hippocampus. So taking that into account, this is one of the many differences that we have noticed from the pathophysiology standpoint between sialidosis and galactosialidosis. And these, of course, differences need to be taken into account, especially considering therapy, because as summarized here, investigational preclinical therapies that we tested in this mouse model gave quite different results. In galactosialidosis, basically everything worked. We have tried BMT very early on. These were one of the early studies of genetically engineered models. We have tried BMT with normal and overexpressing transgenic bone marrow. We have tried the bone marrow-mediated ex vivo gene therapy. We have tried enzyme replacement therapy, and lately AAV-mediated in vivo gene therapy. And I can tell you the characteristic of the enzyme. The enzyme is secreted in circulation as a zymogen and is very readily internalized by different cells and in different tissues, is actually stable in circulation. So this is quite different with when we go to sialidosis, because it's exactly the same approaches shown here for sialidosis mice. Actually, most of them failed. The bone marrow transplantation and bone marrow-mediated ex vivo gene therapy failed because the bone marrow could not engraft. Enzyme replacement therapy failed because the enzyme is extremely immunogenic. So we going back to some of the talks that we heard yesterday, maybe encapsulating the enzyme into, for instance, natural vesicles like exosomes might be a way to go if we want to go into that direction. The one that holds some promise is AAV-mediated in vivo gene therapy, and not to forget unconventional therapies that I will touch upon in a few minutes. So I go very quickly through the preclinical trials for GS. As I said, they were very rewarding in a way and successful. So we tried an AAV-mediated therapy using a 2A vector that expressed the enzyme exclusively in the liver. As you heard yesterday from Alberto Auricchio, is the same concept, the same type of vector. The only difference is that ours is self-complementary vector. We tried a minimal dose of vector needed to reverse the systemic phenotype, the time of injection, and I will just briefly show you the results of the long-term toxicity studies with very high dose of vector. With this, in these studies, we of course follow the mice for up to a year post-injection, single intravenous injection, and you see that indeed you see expression of the cathepsin A, prolonged expression of the cathepsin A up to a year post-injection, and this is of course accompanied by a resolution of the oligosacchariduria as a marker for assessing the effect of therapy. And the other thing that we are often doing is to draw blood from the mice and use the serum from the injected mice to correct disease patient fibroblast. As you see, with 3 independent mouse sera, we can correct both cathepsin A as well as Nurr1 activity in galactosialidosis.
Alessandro,
yes?
Your time is almost over. Please, can you go fast with your slides? Yes.
Well, thank you very much for telling me. I try to run through this. I think the others went over time, but never mind. So the AAV-mediated gene therapy for sialidosis is using a similar construct to give quite promising results. I show here the resolution of oligosacchariduria, expression of the protein in different mouse tissues, including the brain, and most importantly, the amelioration of the hippocampal pathology. We can reverse some of the effect of the Nurr1 deficiency in terms of accumulation of APP, APP positive plaques. As you see, there is a decrease in the APP accumulation, and this is the quantification. Based on these results, we also tried the Nurr1 expression in AD models. We used the 5-time FAD to see if indeed the expression, overexpression of Nurr1 could have an effect. You see that intracerebral injection of AAV Nurr1 in combination with this auxiliary protein PPCA is very effective in the hippocampal region, and this has a dramatic effect on the accumulation of plaques in the 5-time FAD model. So the AAV-mediated therapy could be really a potential, I would call it choice of therapy for both galactosialidosis and sialidosis. And I will finish by addressing sialidosis type 1, because this turned out to be a large number of patients. We have over 30 patients that have been referred to my lab, and there are several others in Italy, in the Netherlands, in several parts of Europe, South America, and India, and so on. So this is a larger population of patients that are in fact mostly misdiagnosed because some of their features resemble other conditions. I skip over this because this has been published. We have tried to use PPCA as in a preclinical mouse model of sialidosis type 1 to increment the residual activity of Nurr1. And that was, I think, in a way successful. But as I mentioned, AAV-mediated in vivo gene therapy seemed to be really the therapy of choice for galactosialidosis and sialidosis. We are going to test this in large animal models so that we are generating collaboration. Actually, I'm not generating, is Heather Gray Edwards in Boston, together with Miguel Esteves are generating CRISPR-Cas9 U1 knockout sheep that we will use eventually to test AAV-mediated therapy in a larger model. Last but not least, I think we all need to take into account daily habits that can slow down the progression of the disease and ameliorate disease symptoms. So that includes the diet, food supplements, physical exercise, as well as medications. And we have experimented with a couple of compounds that are actually over-the-counter available, like beta-in and tanganil, which are modified amino acids that manage actually to increase slightly the residual activity in a type 1 sialidosis fibroblast from patient. And because of that, we have tried in a large cohort of sialidosis type 1 patients that seem to, some of them at least, seem to respond to this type of alternative therapy. And we have even tested this in a mouse model, testing both NDO1 activity in kidney as well as in white blood cells, as well as reduction of urinary oligosaccharides. So this type of combination of approaches and seem to at least be useful to ameliorate the overall quality of life of patients while they are patiently waiting for us to develop target therapy. And again, this is a very, I think, indicative image. I think a small grain of sand make eventually a mountain. So we are making slow progress, seem to be slow progress, but in fact, we have made many progresses, especially in the last decades. I am going to thank my people who have contributed tremendously to this work, but primarily the patients. This is Daniel, at sialidosis type 1, and meet Lauren, one of the galactosialidosis patients who I've been following for many, many years. I've been to her graduation, and I acknowledge all my collaborators as St. Jude and outside St. Jude. And I thank you very much for your attention.
Thank you very much, Alessandra. I'm sorry that no questions are allowed anymore because of the lack of time, but I think your email is very well known, and participants who have a question can send you an email and get an answer from you. Thank you very much. And now I will close this session and we'll go over to the next session. Thank you very much, all the speakers.
Thank you.